thanks to each of you for joining this afternoon. Um, One of the things that I really enjoy most about our office is finding opportunities like this to bring to our faculty across the health sciences. And it's always wonderful when it comes together so beautifully to highlight the talents already on campus. A lot of times we'll be speaking with organizations outside of the university as how to connect, but this is very special and very unique because this is going to give you some insights into the many strengths out of the School of Engineering and Applied Sciences. And I have to say, Dr. Zhuang, since he stepped into his role, I think about a year and a half, 2 years ago now,
Yeah.
has been such a wonderful collaborator and willing to do these talks as we've requested. So, obviously he is the Associate Dean for Research in the School of Engineering and Applied Sciences, but at the same time, he is also a professor in Industrial and Systems Engineering and is wearing multiple hats on campus. And so he selected his colleagues, which I will let him introduce his colleagues, but I know all of them and appreciate the work and all the things that they're doing to try and find collaborative opportunities across the health sciences and across the university. So I thank them for taking the time to prepare for this. And obviously, there's a great deal of interest and hopefully future conversations can come from this. Lastly, I'll say the talk is recorded. If you have colleagues who might have missed it, I will put the link to our Health Sciences YouTube channel in the chat, but the talk will be posted there. I will let Dr. Zhuang decide how he wants to take questions, but I can help with moderating those. But I would assume that you'd like to save them more towards the end once everybody's had a chance to speak, correct?
That's correct.
Okay. Thank you again.
Okay, thank you very much, Jill, and thank you very much for everyone for joining. I know it's a Monday after the holiday breaks, and everyone have a lot of other options. So today we're very happy to bring a group of very talented, gifted professors from the School of Engineering and Applied Sciences, and we're going to talk about our own experiences, including like applying AIs to health science and for innovation. But what's most important is we're looking for the future. We're looking for the future collaborations. We're looking for the future ideas, and we want really to bring in a group of talented people to work together for at this university and beyond. So we're going to bring a four panelists. I think all of them has the camera on. Laura Cavuto is a professor and director of undergraduate studies from Department of Industrial and Systems Engineering. Professor Yun Wu is a professor and chair at the Department of Medical Engineering. And Professor Jun Xia is a professor and director of graduate studies at the Department of Biomedical Engineering. And last but not least, Professor Jin Junxiong. He is a SUNY Emperor Innovation Professor and the Professor of Department of Computer Science and Engineering, and also Director of the Institute for Artificial Intelligence and Data Science. I'm going to introduce very briefly of who I am. So I'm, as Jill mentioned, that I'm associate for research at School of Engineering and Applied Sciences. I'm also a Morton C. Frank Professor, Department of Industrial and Systems Engineering. So I've been UB for about 17 years. So for the C's, School of Engineering and Applied Sciences, at a glance, we have about 10 departments. With newest department is AI and Society department. We also have about 5,000 undergraduate students, more than 2,000 graduate students, about 300 faculty members, and our annual research expenditure is about $100 million research expenditures. So at UB, I'm directing a lab called Decision Risk and Data Lab. So I've been integrating operations research, big data analytics, game theory, and decision analysis to improve the mitigation, preparedness, response, and recovery from natural and man-made disasters. But I am also interested in other applications to healthcare and sport and transportation and so on. So recently been working a few projects funded by Department of Homeland Security, including the public safety problems, border patrol problems, and wildfire management problems funded by National Science Foundation. Actually, I have a few joint proposals with Jacob Schools of Medicine on the healthcare cost side of the wildfire management side. and recently working on the Arctic projects, try to understand with climate changing, what's going on in the Arctic and in Alaska and so on. So without further ado, I'm going to turn to Laura Caboto. Each of the panelists will have about 5 minutes to introduce their research backgrounds, and then in the end, we're going to have a panel discussion. Laura.
Perfect. Thank you, June, and nice to be here with everyone today and the other great panelists. So my work in industrial and systems engineering is in the area of human factors and ergonomics. And I have been at UB for about 13 years in covering a range of applications within the human factors area. Next slide, June. So the main thing that we have focused on through some projects with the Department of Surgery and the Department of Emergency Medicine is the application of AI and objective metrics for clinical skills assessment. So as many of you may know in medical training, it's often a subjective assessment by an expert who has watched the individual perform a task multiple times. But this is often time consuming and a challenge on resources. And so we've been looking at ways in which we can better support effective and efficient training of clinicians, in particular, in this case, surgeons and emergency medicine professionals. And so we've looked at brain imaging, eye tracking, and other physiological measures. And then the piece where AI comes into play in this. So we've used some, traditional machine learning and deep learning models for quantifying skill level. But then the piece that we're working towards now in this space is how do we provide feedback in this, right? So if we can assess the level of skill that someone has, how do we, help that trainee in knowing what they need to improve? And so we've been looking at ways to provide either video or other modality-based feedback so that we can do that maybe in real time. So as an individual is practicing a task, we're collecting some of these metrics. Can we use those pieces to share that information back? And what does that need to look like? So where the human factors comes into play here is what are the best modalities for presenting that information? When should we present it, in the middle of a task or after the task? And how do we, support motor learning and cognitive learning? So learning of knowledge as well as learning of the motor skill, because we still need the practice to develop those motor skills. Next slide. Another piece that we've been working on now in terms of AI application into surgery in real time. It's also what do people, what do clinicians actually want from AI support in their clinical practice? And so we, at one of the large surgical conferences, looked at how to, or asked surgeons, right, what do they want and need, right? And so we've been analyzing that from a qualitative perspective to say, what are those needs and how do they differ based on the level of skill and expertise? So attendings versus residents. And in this case, right, we saw that, attendings had more preference for maybe some specific identification, whereas trainees really valued more of the educational support that an AI tool could provide. Animation, June? And so you can see here from a few quotes, right, in looking at a resident versus an attending, we could see that the resident here wanted some decision support. So how can we provide them with effective real-time decision support that they can use in the middle of a case? Whereas the attending just wanted some basic maybe identification and knowing, what might be a danger area for them and what should they look out for in order to manage an injury. Next. Right. And so we're looking at, and this is an AI generated image, but looking at ways in which you might be able to provide this information back. So, what can we tell someone in the middle of a case about, what risks they might be, experiencing and how should they respond or how should they react? And does that need to be customized or adapted based on the level of skill of the individual performing the case? Next. And then in patient care, we recently received a new grant from NIDLR on rehabilitation research, where we're looking at real-time support for patients in home-based rehabilitation. So push many patients in rehabilitation, particularly in this case post-stroke, can't get to a therapy facility regularly. And so we're looking at how can we provide them with real-time support from a combination of maybe a clinician plus, an AI agent. And how can we help them in progressing so that they maintain their rehabilitation programs? Because it's often, the case that individuals stop their rehab programs when they're sent home without any support. And so this collaboration with rehabilitation sciences, computer science, and then the Idea Center in School of Architecture, and then collaborators down at Georgia State in rehabilitation as well. And so we're looking across the spectrum of how can we use these agents and AI tools to support where they're needed. So providing them in an effective way and a way that's going to be meaningful for both clinicians and patients.
Thank you, Laura. Yun?
So thank you very much for the invitation. It's my great pleasure to share my research with you, and then looking for collaborations in the future. So my research primarily focused on nanomedicine by nanoparticles. So we use nanoparticles for cancer screening, early detection, and therapy. Next, please. So I have two research areas in my lab. My lab is engineered theranostics lab. So on the left, you can see our primary research focus on in vitro diagnostics. So basically, we are interested in developing new biomarkers for cancer screening, early detection, treatment response monitoring, surveillance, and prognosis. So this is more like a blood-based test for cancer patient care. On the right-hand side, this is a second research direction. We're interested in developing new nanoparticle-based therapeutics for cancer treatment. So in my lab, we focus on lipid nanoparticles and also polymeric nanoparticles. So we can deliver different types of therapeutic agents, for example, small molecule drugs, nucleic acids. We also can encapsulate imaging reagents in the nanoparticle for diagnosis and therapy together. On the surface of the particle, we can decorate the particle with targeting molecules to achieve cancer-targeted delivery. Next. So for the first research direction, we're particularly interested in exosome-based in vitro diagnostic assays. So exosomes are small extracellular vesicles released by cancer cells. On the left-hand side, you can see a photo of the exosomes. So basically, we are looking at exosomal proteins, RNAs, and also carbohydrates for cancer patient care. And then currently, I'm working with clinicians and also statisticians from Roswell Park Comprehensive Cancer Center, Mayo Clinic, and a small company for Robin to develop the different types of liquid biopsy tests for lung cancer, breast cancer, colon cancer, prostate cancer, ovarian cancer, either early detection or treatment response monitoring. Next, please. So these are different types of assays developed in my lab in the past about 10 years. They have different functions. So on the upper left corner, so you will see tethered cationic lipoplex nanoparticle TCLN biochip. So this biochip can be used to detect exosomal RNA biomarkers. And then we also developed the microfluidic cationic lipoplex nanoparticle MCLN biochip to reduce the assay time from hours to 15 minutes. But this assay is also for exosomal RNA detection. For TCLN and MCLN assays, we can detect exosomal RNA from all exosomes in the blood. If you are interested in a subpopulation of exosomes, for example, tumor-derived exosomes, we developed this immunocell biochip, which is in the middle upper section. So for this immunocell biochip, we can detect RNA in a subpopulation of exosomes. So depending on the antibody we used, so we can target tumor-derived exosomes. We can also look at immune system-derived exosome markers. And then recently we built a surface plasma resonance-based biosensor to measure exosome protein markers. And then we integrate the immunocell biochip together with the surface plasma resonance biosensor together, and then develop this exosome protein RNA one-stop ExoPros biosensor, which is shown on the right-hand side. So basically, for this ExoPros biosensor, we're able to capture a subpopulation of the exosomes, for example, tumor-derived exosomes, and then we can detect protein, RNA, two types of biomarkers simultaneously with this assay. Next, please. So this is just the example data. We use the ExoPros assay to detect lung cancer. On the left-hand side, you can see we have on the x-axis, we have the protein marker EGFR. On the y-axis, we have the RNA marker miR-21. And then we have three groups of patients, low risk, healthy controls, and also patients at high risk for lung cancer and lung cancer patients. So basically, the blue and the purple are the healthy controls, and the orange dots are lung cancer patients. So basically, you can see we have a pretty good separation between these two groups, healthy versus cancer. And then on the right-hand side, we have a second pair of protein RNA marker, which are LG3BP and the miR-210. And then similarly, you can see the healthy controls, they are basically at the lower left corner, and then cancer patients, they move toward the upper left corner. So upper right corner, sorry. So basically, you can see in the table, when we have individual biomarkers, their diagnostic performance varies. However, when we combine all markers together, we achieve a very high diagnostic sensitivity, specificity, and accuracy. So we're close to basically 100% accuracy. Next, please. And then the second research direction focusing on cancer therapy. So I collaborate with Dr. Chong Cheng in chemical engineering, and then we're working on sweat ionic polymer-based nanoparticles to co-deliver paclitaxel, gemcitabine for pancreatic cancer. So far, we're still optimizing this sweat ionic polymer-based drug delivery system. And then next. Can you click one more time? Okay, thank you. So basically, the other type of nanoparticle is lipid nanoparticles. So I have been collaborating with Dr. Pam Hirschberger to develop lipid nanoparticles for EGFR TKI resistant lung cancer treatment. And then on the right, you can see with tumor targeted particles. So basically, we see the particles are primarily accumulated in the tumor site. So the tumor site is indicated on the MRI images with the yellow cycle. Without the targeting, so basically the particle will quickly be cleared out by the liver, kidney, spleen systems. And then I also collaborate with Dr. Stacy Blaine to develop ER/PR HER2, develop this. This is a lipid nanoparticle to deliver out peptide. The out peptide is a new therapeutics for ER/PR positive, HER2 negative breast cancer. And then at the bottom, I also try to expand my research from cancer to other diseases. So I've been collaborating with Dr. Rogang Zhao in biomedical engineering department to develop these lipid nanoparticles for pulmonary fibrosis treatment. And then on the lower right corner, you will see with our nanoparticle treatment, the fibrosis actually can be inhibited. And then next, please. So basically, this is a summary. We have a liquid biopsy test for cancer patients and also healthy people like us. Our goal is try to detect cancer at early stage, and then for cancer patients, we try to develop new therapeutics. So far we're looking forward to collaborating with people with AI expertise to help us. For example, for cancer liquid biopsy, the patient clinical data is very sometimes it's very messy, and then we try to combine or develop this like a statistical model, try to integrate all clinical data, including the biomarker data together to develop a better diagnostic system. And then for cancer treatment, we're looking for AI-assisted biomaterial design, and then help us better develop a drug delivery system for cancer. Thank you.
Thank you very much, Jun. Jun,
All
you're
right,
next.
sure. Yeah, thank you. It's my pleasure to share my work here. So I'm also in the biomedical engineering department, and I have been here for like 11 years. Next slides, please. Next one. So quick overview. So my lab is mainly work on photoacoustic and ultrasound imaging. So ultrasound imaging is very popular. Everyone knows it has been used widely in the clinic for visualizing structures inside the tissue. However, there are some issues. For instance, ultrasound cannot visualize the color of the tissue. It can only give you the acoustic reflection. Second, the image quality is operator dependent. So that means if you have a very skilled ultrasound operator versus a brand new, like a newly graduate ultrasound operator, the image quality might be different. Next, please. So that's why we're working on a new imaging technique called photoacoustic imaging to visualize color inside the tissue. So we consider it to be a kind of add-on to ultrasound imaging. So the principle is we shine pulse laser light into a tissue, then anything that absorbs light will generate heat, generate heat, and the heat will create expansion. The expansion will generate ultrasound waves that can be detected by an ultrasound transducer. So we still use the same ultrasound transducer, but in addition, we add a laser, short pulse laser, which is very safe to generate a colorful image inside the tissue. Next, please. So by using this, we can also integrate automated ultrasound scanning into the system. So in this case, we can get rid of the operator dependence, because using automated scanning, you can get an entire picture of the human tissue. You don't need to rely on experienced operator to identify where the suspicious lesion is. You just get a complete 3D volume, and then we can combine with AI techniques to provide a more accurate diagnostic. Okay, next, please. So one of the research we're working is breast cancer imaging. This is in collaboration with Roswell Park Cancer Center. So the animation shows imaging system we conducted, we built in the lab and then moved to Roswell Park. So we have a imaging room there to image patients. So the image showing on the right is the vascular structure of a patient with breast cancer. So at roughly the top right corner, you see a dark region, and then there are all the vessels is growing toward that region. That's a feeding vessel of the tumor. So using this feature, we can identify like the malignant lesions inside the breast. Because one of the main issue for ultrasound breast cancer imaging is a high false positive rate. So that means it will identify lots of suspicious regions, but many of these are actually benign. You don't really need to do biopsy. By looking at the vascular patterns around the suspicious region, you can identify whether it's benign or malignant. Next, please. So another application we use is adding photoacoustic and automatic scan for biometric application. So the image showing here is a vascular structure from different actually students at UB. So you can see different human subjects have different vascular structures. And based on these vascular patterns, we can actually use it to identify different different person, and it's considered to be more secure compared with fingerprint or face imaging. Okay, next, please. So another clinical application we're conducting is foot ulcer imaging. So we know when people are getting older, so there will be foot ulcer. And these ulcers won't heal for a long time. They are considered as chronic foot ulcers. Most of these patients will be seen at the vascular clinic to restore perfusion to the vascular structure. So one issue with vascular surgeon is after surgery, it's hard to tell whether the perfusion to the tissue has been restored or not. So we're using this photoacoustic system to see the colors in the tissue. So what gives you the color is basically the blood vascular structures, because hemoglobin in the vessel will give you the color compared with the other tissues. So by looking at the pattern, so the top image is a subject with a good perfusion. You can see the clear vascular pattern. And then the bottom image is a subject with a poor perfusion. So you can see the vascular pattern is blurry and distorted. Okay, so next, please. So with this clinical collaboration, we mainly work with Roswell Park and Windsor Radiology to image breast cancer patients. We have imaged roughly like 250 breast cancer patients using the system shown on the top left corner. And then for the ulcer imaging, we mainly work with UBMD surgery clinic, particularly the vascular surgery clinic, which is in the Maple Mere location. We have imaged around like 150 patients so far. Next, please. So another goal is to, besides the system development, we really want to see how can we integrate like AI with the imaging system to improve the diagnosis. So an example shown here is the AI-based image enhancement. So the original image showing acquired from our system is a little bit blurry because of the system noise, thermal noise, electronic noise. But by training a 3D UNet network with ground truth data, we can clear up the image to remove all the background noise and enhance the vascular structures. So the output image shows a 3D UNet trend model, how it can improve the vascular structures of the human subject. Next, please. So another issue with anything related to optical imaging is the skin color. So we know for a subject with a darker skin, the skin will absorb light because anything dark will absorb light. So a major challenge is with these optical techniques, how can you see vascular structures under a darker skin? So again, we use a AI trend model. So we basically ask students to manually label the skin on 3D image, and we train on like a few thousand slices to label the skin. And then we use a trend model to automatically remove the skin layer. So from the input, you can see the skin layer gives you a very strong signal. You cannot really see the vascular structure underneath. But by using the skin removal network, you can remove the skin layer in 3D and then reveal all the vascular structures underneath. Next, please. And besides the imaging enhancement, we also use AI to improve the diagnosis. So the example shown here is the breast cancer imaging. So from the breast cancer imaging, we get a 3D view of the vascular structure of the breast and also a 3D view of the ultrasound, because photoacoustic is an add-on to ultrasound. We still get the ultrasound image. And by using an AI model, we can analyze all these data in 3D. We basically divide this data into like multiple chunks and analyze one by one and to identify where is the most suspicious region. So the heat map at the bottom right shows a red color region. So that's the network identified suspicious region of the tumor. And then on the top right, we see the cross-sectional image. One is we call it acquired ultrasound image, is an image captured by our imaging system. And then the clinical ultrasound is the image captured by the Roswell Park's commercial ultrasound system. So we draw a cross line across a region highlighted by the heat map. We can see the tumor region matches very well with the clinical ultrasound. So this is kind of a proof that we can use AI kind of AI techniques to analyze multi-modal data for the photoacoustic ultrasound and to identify the breast cancer. Next slide, please. So for the food ulcer imaging, we're also using AI-based approach. It's called a radiomics approach. Radiomics is a technique to identify like different image-based features. Some of the features is impossible to analyze by human. So experienced radiologists know these features, okay, but it's very hard for new radiologists to be trained. So we use radiomics library to extract like thousands of different features from patients, some patients with good perfusion, some patients with poor perfusion, and then we combine all this data into training data sets. And after we train on thousands of radiomic features, we identify the top like 12 features. And then based on these top 12 features, we can use a model to predict whether this patient have a good perfusion or a bad perfusion. So this will really help with the clinical reading, because otherwise, by looking at the vascular image itself, it could be sometimes hard to interpret which one have a good or bad perfusion. Okay, next, please. So in summary, right now we work mainly with Roswell Park and Windsor Radiology and the UB Vascular Surgery Clinic. These are the clinicians currently helping us to recruit patients. Were very helpful, and we look forward to explore new collaborations with a medical clinic. Thank you.
Thank you very much, Jun. Last but not least, Jinjun.
Okay, good afternoon, everyone. Thank you so much for having me here. And so my topic is called Earth AI for the health sciences cross-disciplinary research, right? And most of you know I'm a AI researcher, and for me, I'm really promoting to use AI to solve the domain problems, and by working with the domain experts like yourself, and how to identify the real research challenges, and that we can use AI to help. So I'm going to illustrate a few examples and connect with some of the early speakers' research work as well. So next page. Right, so most of you know, like one of the projects I have at UB is this the National AI Institute for Exceptional Education, where we use AI technology to do the screening of children who may need the special education needs, and also then apply the AI to do the enable the universal screening and also for the intervention. So tie back to like Yun's work, right? Like a lot of this medical cancer screening and also the treatment. So in this same sense here, this is only for the speech and the language screening and the treatment. So Jim, sorry, I just can click a few times. Okay, yes. So these are all the AIs. A few more, please. So you can see I'm developing the AI technology, right, to enable the universal screening and also the treatment, but for the special education needs children. And so this is kind of for one of the biggest projects I have here. But as you can see, in order for me to identify this problem, right, I have to talk to special educators and the patients as well, and also the parents who are taking care of the patients. So that's how we identified that the speech and language screening and treatment is one of the biggest challenges in our school system. And taking a similar approach, I've been working with a number of other colleagues here at the UB and applying the AI technologies to help them to solve some of the domain challenges as well. As you can see, for all of these use cases, right, and the core is very similar. And how do you understand different modality of information, because the speech and language, right, which means the audio information and the natural languages, and also the video as well. So now I'm going to show you, okay, how two examples. So next page. So this is also in collaboration with like Dr. Peter King, right, and also the Steve Schwarzberg and the Jeannie Young and a few others at the Department of the Surgery, and about how do we use AI to help with the surgeons and to make better decisions. And Laura, I think your work, right, helped to see, the different attendings and also the residents, right, they may have different needs. One thing is how do you provide the real-time decision support tools and for surgeons while they do the surgeries. And the second one is how to help the surgeons to train the future surgeons. And because of this is a lot of the surgical videos, right now is available, and including the audio files as well. But unfortunately, this video and audio files are typically large, right? And also the very complex, requires deep domain expertise to understand. And it's very time consuming for us to ask those experts to label the data, right? Even ask them to collect and then annotate the data is very time consuming. So that's why we are working with the Dr. Kim and Schwarzberg, etc., and to develop a foundational AI models to understand the surgical videos. And so starting from how do we help the doctors, right, to annotate the data better and easier, and also with limited annotation, because annotation takes time, we use AI to be able to produce more high quality data with the different labels. So that's kind of shown on the left-hand side, right? With that way of to be able to build the data, we are able to train the models. And starting from how do we recognize, right? This is talking about during the surgery, during the surgery, right? And the real-time videos streaming to the processing unit, and how do we identify different objects, right? And later on we can provide more informed information for the surgeons when they make decisions and what to cut, what not to cut. And this really ties back to Laura's study, right? And how do we provide such a decision supporting tools? And this is still work ongoing, and we already have some very nice results and published and top AI conference. So this shows you how do we develop AI for the multi-modality understanding, right, for the surgical videos. The next one, please. So this is another one. It's about more focus on the natural language processing, and this is again in collaboration with our colleagues Teresa Cochran and Andy and Renee and a few others, and in the CTSI. And this is about how do we make the medical documents simplified, more accessible for a general public. As you know, right now, okay, most people, right, like 1 in 3 American adults, they use online tools and to look up for the medical information. And a lot of the medical information is in the of course, scientific papers, right? And even though they are very accurate and a lot of very informative, but because of the complexity and a lot of the domain jargons, right, made it very difficult for the public to consume and access such information. So we developed a project around this challenge. We call it Med Simplify, and it's an agentic framework, okay, method to simplify the medical documentation. And so as probably you cannot see, just you can think of different agent, means different AI components. And so in this case, we use a different term agent and the planning agent, generation agent, and the evaluation and the memory agent. It's a multiple agents working together and come with this simplified medical documentation. And then you may say, okay, there's already a lot of work already, right, in the literature. But so we take our approach and compare against different techniques available right now, and also leverage our colleagues' connections to the community. And we did a human studies. And at the bottom, the table shows the results, right? And our method compared to the state of the art, medical text simplification, and not based on some called metrics, only like a human the AI understand. All of this metric in terms of the preferences, right? Readability, clarity, and the completeness is all based on the humans, community members' assessment. And as you can see here, and our method consistently better, consider beat the state-of-the-art method. So this is something really out of this good collaboration with our colleagues at the CTSI. Okay, so this is a kind of 2 examples. And so really, I'm really promoting, right, people who are developing technology to work with the domain experts, identify the challenges, and make sure our technology can make a difference and for our society. Thank you.
Thank you very much, Jingjing. So this is our last slide. Now I think we're going to open to some of the panel discussions. I'd have a few like pre-popped questions for the panelists to speak on. But if any of the audience have any questions, feel free to type in the chat box. And as you see, we're on the right side. We also have a post a panel survey. It's only take like 2 or 3 minutes for you to fill. I'm also going to put that in the chat box, especially if you're looking for collaborators in the engineering, or if you're an engineer looking for collaborators in the medical fields. Feel free to put that in the survey so that we can match you. That's the purpose of this conversations. Now I'm going to open to the panel and see who want to go first with any of the questions you want to uh, have comments on.
So June, we just randomly answer any questions, or what is the format?
Jinjin, what's your favorite question to answer, please?
Oh my goodness. Okay, so I can take the first one, right? So, and I think this is cross-disciplinary collaboration is something that's very important. And even though we all recognize the importance of the cross-disciplinary collaboration, but in reality is, as you mentioned here, there's a lot of challenges, right? Because people has different priorities, right? And even for publications, they have different choices of venues for publication, right? And one publication in one domain may not be helping the others, to be recognized. So that's one thing. Yes, there's a lot of challenges, but I think a good collaboration is really the open mind, right? Make sure that everyone, you need to listen well, right? And from the other party, what are the real challenges, and also try to learn the terminologies. And there's a gap of terminologies as well, right? So you need to be patient, open-minded to understand the process. And especially I think for the engineer, from the engineering side, we think we have the technology, but sometimes we can be easily feel fault like the other party, right? And from the domains, they cannot formulate the problem. I mean, the problem is too trivial, right? And too easy, and it's not easy, it's not helping me to advance my kind of academic depth. But in fact, I find like a lot of the domain challenges, right? Once you start to solve it, you will realize there's a huge gap and a huge need for technology advancement. So really, probably my suggestion is really more from the engineering side, for any engineers, right? Any of the researchers who want to help the domain experts, please keep it open, keep an open mind, right? Listen to their description of the domain challenges, and to understand their terminologies. And I think only by doing so you can get a very fruitful collaboration, and also solving some interesting problems too.
Thank you very much, Jinjin. Jinjin, actually, we have a question for you in the chat box. So is the Math Simplify available for UB faculty and students?
I think so. We can make it available. Yes, right. Okay, it's still like kind of with our the tools with the data that we have, but we'd be happy if anyone interested to send us some data, and we can test it out, and we can probably make it as a service, so people can use it as well.
Great.
Yeah, Diana, yes, please follow up, send an email to me, if you're interested, have any articles you want to try, and I'd be happy to test it out, and we can see how we can make it as a kind of a tool, right, as a service, so you can try it out, and to help more colleagues as well.
Great.
Yeah.
Laura, do you have any questions that you feel passionate concerning? Maybe I'll
Yeah,
human.
sure. So I'll talk about the second one since my area is in human factors. And so I think when we've worked with clinicians, I think the workflow piece is an important component in how to fit these pieces into the workflow. And so I think that's where it requires, as Jinjun was talking about, a lot of discussion and talking amongst the parties. So having systems that are built with the clinician involvement early, because I think that understanding that work process and what's actually happening in the clinical decision making and who's making clinical decision making, who's making the clinical decisions, I think is critical to making sure the system will actually be useful. For example, I have a PhD student who's working on some explainable AI for patient discharge. And after talking with like 10 residents and social workers and nurses who are, in the discharge process, what he realized is that, there's a lot of other factors that are going to affect discharge regardless of what type of AI system you have, right? And if it gives you a score, what, explainability you give to it. So I think, we really need to make sure that everyone is at the table early, so that we can get those outcomes.
Great. Thank you, Laura.
Mhm.
Yun, you have any questions that you want to discuss?
So I think I can briefly chat about the translational barrier and as well as the education and workforce development. So currently in my lab, I don't do much AI related work, so I probably only can comment on translating technologies from lab to clinic. So basically, I think the major barriers for engineers would be number one is we're developing very, I would say, innovative technologies. And then, however, sometimes when I speak with clinicians, and then they all agree technologies are highly innovative. However, multiple factors have to be considered when we translate these technologies to clinical settings. So, for example, when I develop liquid biopsy assays, and then one type of assay is a microfluidic device-based assay, and then we believe it shortens the assay time from hours to minutes, which will be highly useful and valuable for cancer screening because the large population of the patients needs to be screened. However, when we try to move that to clinical settings, and then it turned out the microfluidic device is really hard to use, not very user-friendly. A lot of technicians in the lab, they are not very familiar with the microfluidics setup, and then even as engineers, we feel it's very straightforward, it's easy, but when we try to move forward and then train new users, and then we feel resistance. So basically for clinical labs, they prefer either liquid handling system, everything is automatic, or a very, very easy user-friendly assay. So basically, I think this is a barrier between engineers and the clinicians, how we move the technology to clinical setting. And then also sometimes we also need to consider, for example, for our assays, we always want to reduce the cost. And then because when we translate the technology to clinics to patients, and then so the cost analysis is also a critical component. The technology is innovative, is useful. However, if it's too costly, and then so basically this is more like the market issue. So who are going to use this very new technology? So that's my 2 cents for the translational barriers I have experienced. And then for the education workforce part, just like to facilitate the collaboration, and then the next generation, I think we have to train them to speak the language that basically for biomedical engineering department, we try to train our students to speak language that not only engineers understand, but also healthcare professionals, they can understand. So for example, in our department, we have been working with people from neurosurgery to have our students go to the operating room to stay there with clinicians and surgeons for a short period. It's like a 1 week clinical immersion program. Students will go to the clinical environment. They will basically work with the medical students, and then they exchange ideas. And then so basically we hope this type of small program can train our students in the clinical relevant environment. And then, so basically what they learned in the classroom will make sense to them. And then they can also be prepared for future positions that requires engineer to work together with healthcare professionals.
Thank you very much, Jun. Jun, you want to comment?
Maybe, yeah, I think the panelists already answered most of the questions. I will just add some of my general thoughts in general to all these questions. So, and also I see in the chat box one there is a question regarding how the research was initiated. Maybe I can briefly mention that. So for our research, it was initiated by emailing the clinicians. I found the most success is for clinicians who are affiliated with Roswell Park or UB. We also try to email clinicians in hospitals with no affiliation, but success rate is lower. Okay, if they have affiliation with UB Roswell Park, it's easier. And also, one challenge we found with translation barrier is clinicians are very busy. So we know they need to handle clinical work. They don't have time to do research. So some of the research was actually initiated from the fellows or residents in the clinic. So we have initial meeting with the clinician, and then they assign their residents or fellow to work with us. So the resident fellows, although they are also very busy, but they are very willing to work on research work or write joint proposals. And we have more frequent meetings with fellow and residents. And once the research is established, we can involve the clinicians to help with patient recruitment, but fellows can do that as well, and for preparing the grant proposals. And for the future opportunity, maybe how UB can help. I think we benefit a lot from some of the UB internal grants. For instance, the CTSI pilot study grant. Our food ulcer imaging was initially funded by that grant, which allows us to collaborate with the clinician, acquire preliminary data for NIH grants. So if there UB have more kind of pilot study grants to combine clinicians with the engineers, that would be very helpful. So so I think those are some of the opportunities, like maybe Junzhuang can explore. Okay, all right, just my 2 cents here.
Thank you, Jun. Sanjay, I see that you have a questions in chat box. It's an excellent question. How can CTSI help for those collaborations? I especially like the second part of the question. Can AI help with that? I think like the meetings like this would be a good starting point to get exposed to like both sides, the engineering side and also the medical side. I think we really need to know each other. We have lots of new faculty actually on this, even on this call right now. They probably even don't know that what is CTSI and what type of collaboration opportunities are there. I think there'll be going to be good opportunity for people to know each other more. Huh? And there are a few other questions in the chat box. And anyone want to take any of the questions? Nora?
I can maybe elaborate a little bit of a Katya's question, right? And how this collaboration originated. I think Dream mentioned some on his point of view, and I think really the collaboration really has to come from both sides. You know, like I just give the 3 examples I had. For example, for the speech language, I have to initiate, right? I have to talk to people. And because I need to write the proposals at that time, right? So I mean, I spend a lot of time to try to reach out to collaborate. On the other hand, right, the other 2 projects, it really is from the on the other side, actually reach out to me, say, hey, I have some of the challenges. Do you think AI can help? Right? But I do spend time to understand, you know, what are the challenge. Is it really interesting or can sometimes some of the challenges can be solved easily, right? Which I didn't mention here. Some of the challenges cannot be easily solved. Then it takes time, which means that you need to willing to listen, but then afterwards you need to follow through as well, right? And some of this collaboration, there's no money involved. In a sense, you have to invest early, and we don't have even the initial pilot grant, right? But out of this outcomes, then we can starting to collaborate to apply for the external grant. I think Laura, you and I also collaborate with Peter, right, and Steve, and as well. So actually, we started to writing the proposals out of that initial collaboration. So I think it really has to both sides need to have the take the initiative. And at the end of the day, it's the time, And like Jim mentioned, a lot of times it's the doctors, right? The clinicians, they are so busy. And if they can spend time with you, right? And so please kind of value their time and try to be patient and listen to their description of the problems.
Thank you, Jingjing. We only have a few minutes left. Any other panelists?
If I can jump in. So first of all, thank you to each of you. I got a lot of personal comments directed towards our office, complimenting the strength of the panel and the time that you all took and how exciting it is to be engaged in the conversations. So thank you for the time for investing with us at lunchtime today and for the time in preparation. One thing I would say is a little heads up. Dr. Zhang and I are working to move forward our third session of Health and AI Symposium, which will be in February of the upcoming year. And so we have a couple of keynote speakers that we're working with right now that look very strong and very positive. So keep your eyes open for that. As part of that, there will be panel discussions. We'll have an opportunity for people to exhibit posters on their current work if they choose to. And so that's another way to continue to make conversation. I would also say, and I, from past experiences, I recognize what it takes to put new teams together. It's an investment, a significant investment. And sometimes things start via opportunities like this and listening to a presenter and reaching out after the fact. But always keep in the back of your mind that offices like mine, the AGRs in each of the schools, we have access to some resources that if you're looking for somebody to find out if somebody on campus is doing work in a particular area and you're unfamiliar, we have some tools that we have access to that we could actually very quickly and easily do some searches for you as far as people who have found funding in a space before, people who have published it with another potential collaborator, and we can do the legwork for you on that. So please know that we're here to be a resource. Is there anything else, Dr. Wong, that you wanted to mention? But otherwise, thank you so much for everything that you all did.
Thank you very much for organizing this. It's a great experience. I hope this is a really good start too. I see lots of potential collaborations already started in the chat box. So looking forward for the brighter future.